Hello and welcome everybody. I'm happy to present the results of our study on effects of drought history, more precisely if and how drought history could impact grassland carbon and nitrogen cycling. Globally, grasslands cover about 40% of the land surface and they have an important role in carbon cycling and carbon sequestration. Grasslands can be highly productive. Under well-watered conditions, plants can be quite active taking up and assimilating CO2 from the atmosphere. And very simplified in this scheme, they allocate this carbon to produce biomass. Plants transfer sugars below ground to produce roots and release newly assimilated carbon, as for example as sugars or acids, into the rhizosphere. In the rhizosphere, microbes can use this labile carbon and can stimulate the decomposition of soil organic matter to release nutrients for plant uptake. And of course, when plants and microbes are active, they also release CO2 back to the atmosphere. Global climate models suggest that some regions will experience strong changes in climate variability. This means that the possibility of more extreme weather could increase. This can increase the number of, for example, extreme hot weather, or increase the number of days without rain and trigger strong changes in rainfall timing. This increase in weather extremes can also enhance the possibility of drought periods in many regions worldwide. For example, in the areas marked yellow or orange on the graph on the right side. Droughts are defined as periods where water loss via evapotranspiration is higher than water supply, through either groundwater access or as precipitation. Basically, droughts are periods of severe hydrological imbalances. Of course, plants and microbes have developed a range of mechanisms to survive droughts. Most plants can regulate their stomata, which can reduce carbon uptake. Plants can also regulate the allocation of assimilated carbon to plant parts that are particularly important to survive droughts. For example, to roots or to mycorrhizal symbionts, to enhance water uptake or osmotic capacity. Also, soil microbes can suffer from water and nutrient deficiency. During droughts, many will reduce their activity and switch to inactive or dormant states, which can change the active microbial community composition. Moreover, as microbial activity is reduced, organic compounds tend to increase during drought. So each extreme drought can trigger several different responses within an ecosystem. Depending on the resistance of the system, effects can be minimal and the system quickly recovers, or disturbances can be strong enough to trigger a change in the state of the system. For example, indicated by species loss, a plant or microbial community change could occur, or finally a change in the ecosystem functioning could occur. So the question of our experiment was, if carbon cycling in a grassland ecosystem that has already experienced several droughts would differ in its sensitivity to another drought, compared to grassland that grew under ambient conditions. We focused particularly on how drought history affects the response of plant biomass and carbon and nitrogen distribution during a further drought, how drought history affects the response of microbial biomass and microbial community composition, and finally, how drought history affects the cycling of newly assimilated carbon during another drought. To address these questions, we conducted an experiment on a grassland site in the Austrian Alps. The grassland was part of a multi-year drought experiment and has been exposed to extreme droughts 3 growing seasons in a row. In each year, rainout shelters were installed and excluded all rain over a period of 10 weeks. Additionally, control plots exposed to ambient weather conditions were maintained. In 2011, after the end of the 3rd rain exclusion period, when rainout shelters were removed and plots were rewatered, basically in the recovery period, we conducted this experiment to study the carbon dynamics in more detail. During this recovery period, a natural drought period occurred, which decreased the soil moisture in the drought history plots, which are the white dots in the right graph, as well as in the ambient control plots. After 10 days of dry conditions, a heavy rainfall event quickly increased the soil water content in both drought history and ambient plots. We tested the effect of drought history on the plant and microbial carbon turnover by using 13CO2, the stable isotope of carbon, as a tracer. After 13C label pulse, we collected above ground and below ground biomass, and we extracted microbial phospholipid fatty acids from the soil. PLFAs, which is the abbreviation for phospholipid fatty acids, are part of microbial cell walls and indicator for special microbial taxa, for example fungi, gram-positive and gram-negative bacteria. Moreover, they contain a lot of carbon and are so a good tool to study uptake of carbon into microbial biomass. Now I'm coming to the results. First, we found that the drought history treatment did not affect the distribution of plant biomass and their response to the drought period. The black bars show the results from the ambient controls and the grey bars show the results from the plots with the drought history. The bars with the red arrows show data from during the drought period. The blue arrows indicate data after the rain pulse. Shoots and roots responded similarly in drought history and ambient plots. Root biomass, however, increased after the end of the drought under more wet conditions. Root respiration significantly decreased during drought in drought history plots, which indicates that these plants may have had more stress during the drought. While the biomass remained stable, we could observe changes in the plant's C/N ratios. The shoots of plants with drought history had a higher C/N ratio caused by a decrease in nitrogen, while the roots had a lower C/N ratio as a consequence of higher nitrogen contents. So, plants in drought history plots apparently shifted their nitrogen allocation pattern. Moving further below ground, we found that microbial biomass was responding very similar to a further drought, regardless of drought history. Again, black bars are the data from the ambient controls and the grey bars are from the drought history plots. Neither before in the recovery period nor during the drought and wet period, the microbial biomass was significantly different from the ambient controls. However, generally only a small proportion of microbes are normally active, so potential changes in the total microbial biomass might not have been detectable. Nonetheless, we found that the microbial community composition was changing in both drought history and ambient plots from the recovery to the drought and the wet period. In the recovery period, before the drought, the microbial community was still different in ambient controls and drought history plots. During the drought period, however, both treatments, the proportion of gram-positive bacteria was increasing similarly, and the overall community composition changed significantly. The rainfall event triggered a further community shift, again similar in controls and plots with drought history. Most strikingly was that the carbon turnover in sites with drought history was significantly reduced and slower during and after the drought period. These two graphs show the dynamics of the 13C label after pulse labeling in plant shoots on the left and root biomass at the right side. Plants in drought history plots, which are shown as white dots, took up significantly less carbon during dry conditions and also turned over the carbon more slowly. This was also reflected in significantly reduced 13C recovered in the fine root biomass at the right picture and also in root respiration, which is not shown here. The 13C recovered was below the detection level. Similarly, the uptake of plant-derived carbon in microbial biomass was reduced in plots with drought history. Most likely, this occurred as a consequence of decreased plant carbon allocation, but could also indicate stronger reduced microbial activity in drought history plots. The rain pulse, indicated by the blue arrow, triggered microbial activity, especially in drought history plots. Here, the precipitation likely stimulated the uptake of accumulated plant carbon for the production of new PLFA stronger than in the ambient controls. Summing up and coming back to our initial question, will grassland with drought history respond similarly to drought as ambient grassland? We found that plant biomass and microbial community composition responded very similar to drought independent of drought history. Plant nitrogen contents as well as carbon uptake, allocation and root activities seem to be more sensitive to drought history. Moreover, microbial turnover of recent plant carbon during droughts was changed by drought history. This indicates that repeatedly occurring droughts can induce a gap between structural and functional responses of the microbial community composition. Overall, we concluded that the projected increases in the frequency of drought periods could in the long term alter the quantity and quality of plant inputs for microbial decomposition and so induce changes in microbial functioning, and this could affect ecosystem carbon turnover and storage. Thank you for watching this video. Goodbye and take care.